Okay, team, we are going to finish up the unit today with meiosis. And I'll just pop this PowerPoint open just to remind you we're in cell cycle mitosis and meiosis. This information plus the cellular energy PowerPoint will cover the material in the first test. I'm sorry, the second test rather. So we made it through most of this slideshow. Remember the cell cycle, haploid and diploid. This is going to become relevant now because we're going to see how in meiosis we take one diploid cell and we make 4 haploid cells. Remember haploid cells, these are the gametes, these are the sperm and the eggs in us. Some organisms have different types of life cycles, but when you're thinking about us, the only haploid cells are the sperm and eggs. Everything else is diploid. Sperm and eggs, how do you get from diploid to haploid? Meiosis. That's what we're going to do today. We already did mitosis. Just to review, it's asexual reproduction. One cell makes 2. It doesn't change the ploidy, so haploid cells can do mitosis, diploid cells can do mitosis, and it doesn't change the ploidy. And then we walked through the phases, which I won't elaborate on now. We've done all this. Okay, so we talked about sexual reproduction, and meiosis is the process which reduces the number of chromosomes by 1/2 in the cell. So it should make sense then that only diploid cells can undergo meiosis. A haploid cell doesn't have any homologous chromosomes to pull apart. Okay. And we're going to see how one diploid cell actually produces 4 haploid cells instead of just 2. We're going to go through all this process with you in just a moment. Okay. All right, so let's remind ourselves about some of the terminology just in a chromosome, just for fun. This is a replicated chromosome. If I draw another one beside it, then we have a pair of replicated chromosomes. We could call this a homologous pair. They have the same genes, right? And then each side consists of chromatid. And remember, the chromatids are the identical pieces that were formed during the synthesis phase of the cell cycle. And then this little spot in the middle that holds them together is the centromere. I think it's very helpful to just keep going over these C words over and over because there's so many of them. Okay. So if you recall, in mitosis, we lined up all the chromosomes along the equator and we pulled the chromatids apart, pulled the 2 identical pieces apart. They ended up in different cells, and that's how we got 2 identical cells, barring any mutation. Okay. All right, so there's just a little review for you. Now, what we're going to do is understand something about meiosis now. And the first thing I have to tell you is that meiosis comes in 2 parts. And so we could talk about meiosis 1 or PMAT1 followed by PMAT2. So we're going to do PMAT1 and then we'll be at the end of that of meiosis 1. And then we're going to do meiosis 2. There we go. And then at the end of that, we should have 4 haploid cells. What are we starting with? 1 diploid cell. We're going to end up with 4 haploids. So here's what it's going to look like. I'm going to do a P, M, A, T. So this is P1, M1, A1, and T1. Okay, and then below that, I'll do P2, M2, A2, and T2. Okay, so here's the deal. Meiosis starts out similar to mitosis in that we have the chromatin condensing to form chromosomes and the nuclear envelope breaking down. So I'm going to draw the nuclear envelope breaking down. In this particular cell, I'm going to use size to indicate homology, okay? And so here's what we're going to do. We're going to have, I'm going to use different colors because there's something that I need to show you, and it's really a pain in my butt to do this, but I'm willing to do it to show you how this works. Okay. So now you can see this is a diploid cell. Each chromosome has a partner. Each chromosome is also replicated, so it consists of two chromatids. Size indicates homology. These two are partners, and these two are partners. And what does 2n equal in this cell? 2n must equal 1, 2, 3, 4. Okay? So here's what's going on in prophase 1. A lot of stuff is going on. We start to see the distinct chromosomes forming from the thread-like chromatin. The nuclear envelope breaks down. And then what happens is these chromosomes find their partner because they're all just kind of scattered about at first. And so they literally go around and they find their partner. How do they know their partners? They have the same genes in the same order. We said that before. Okay. So it's pretty cool. I mean, imagine in a human, there's 23 pairs of chromosomes. So there's 23 pairs of chromosomes, and they literally go around in the cell and kind of start doing their little dance with each other. It's very cute. And then the chromosomes kind of get intimate with one another, and this amazing thing happens in prophase 1 called crossing over. And I want to explain what happens. The chromosomes start to wrap around each other, the homologous chromosomes, and then, believe it or not, they shuffle their genes back and forth. Now think about the implications of this. Where did these chromosomes come from in the first place? One came from your daddy and one came from your mama. In crossing over, your mother's and your father's genes get shuffled around between the chromosomes. And they call it crossing over because literally it's like this. They literally cross little pieces over and then they trade them. And I know this sounds really bizarre, but this is what happens. I'm just showing one crossing over event right here. And so then the result of that, though, is something like this. I guess it would actually have been over there. Sorry, let's put it on the right side. That would actually be red still. Okay, so look what they've done. They've traded genes. Now just imagine the implications of this. Your daddy and your mama's chromosome. Let's say this is the eye color gene and your dad has black hair and brown eyes and your mom has blonde hair and blue eyes. If they trade those genes with one another, now your daddy's chromosomes still got black hair but blue eyes, and your mother's chromosome has blonde hair but brown eyes. And so this happens a bunch of times over all the chromosomes, and what it's doing is it's creating brand new genetic complements of DNA, right, by shuffling those genes together. So it's pretty amazing to think about. I'll draw the crossing over event here just so we can keep track, and it is a chore for me to do that, but I'll do my best. Okay, I'll go back and add the colors in. Okay, so that's prophase 1. A lot of stuff going on. Nuclear envelope breaks down, chromosomes condense, the chromatin condenses to chromosomes, they find their partner, and then crossing over occurs. In metaphase, now I want you to look how these things are going to line up now, because this is different. What colors did I use? Green and blue. Okay, now look at how they line up and compare that to metaphase of mitosis. In metaphase 1 of meiosis, the chromosomes don't all line up along the equator. They line up opposite their partner, okay? And so compare that just real quick, and then I'll erase this because I just want you to remember. If this was metaphase of mitosis, they would have lined up like this, right? They would have all lined up in a big long line. The chromatids got pulled apart. Now they're lining up opposite one another, which is totally different. This is one of the main differences between the two when you look at it in a drawing. Okay, you know this is meiosis by the way they line up in metaphase. Let me do my colors. Let me add a little piece of black there to show the crossing over event. We have spindle fiber formation. So the spindle fibers grow down just like they did before. But now look what gets pulled apart in anaphase. Not the chromatids, but the homologous chromosomes get pulled apart. Oh, wrong color. Okay. Blue one there and a green one. Okay. Draw the spindle fibers in there. So now look, this one's partner is over here. And this one's partner is over here. And now in telophase 1, check this out. 2 new nuclear envelopes begin to form around the 2 new complements of chromosomes, and check it out. Ah, come on. Uh-oh. There we go. Green one up here. Wrong color blue. And blue one in there. Now check it out, y'all. Check it out. What have we done? We've created 2 haploid cells, correct? We've created 2 haploid cells from 1 diploid cell. Notice this. Now we can do cytokinesis here. Let's go ahead and do that. Draw a membrane there. Draw in the nuclear envelopes. And forgive me for not doing the colors in the last drawing, but you get the idea, okay? So in cytokinesis, we divvy up the cytoplasm. Check it out. Now we have 2 haploid cells. Look at these cells. Yes, they are replicated. The chromosomes are replicated, but this cell is no longer diploid. It doesn't have 2 of the same size chromosomes there. We're using size to indicate homology, okay? So that is meiosis 1. And the good news for you is that meiosis 2 is just mitosis. It's mitosis occurring simultaneously in both of these cells, typically in a perpendicular plane to the plane that meiosis 1 occurred in. So you've already seen this, but we'll draw it anyway. And it's going to look like this. So we'll do P2, M2, A2, T2, and then cytokinesis 2. And then there's going to be our 4 cells. Okay, so what's happening here? Well, now we got these 2 cells, and they're going to do what is basically mitosis. And so now the equator becomes in this plane, sort of perpendicular to the first plane. And In prophase, the nuclear envelopes break down. I shouldn't have drawn that in there yet. Sorry, hang on. It's a little too confusing with all those lines. Okay. So nuclear envelopes break down just like they did before. I'm not going to do the colors, y'all, until the very last one, and then I'll show you the colors. Okay. Now look how they line up. Big one and a little one. Doesn't matter if you what order you put them in, big and little, or little and big. It's random the way they all line up. Now look what's going to get pulled apart in anaphase. Remember, this is just mitosis. It's just happening in both of these cells at the same time. So in anaphase now, it's the chromatids that get pulled apart. We got a little one and a big one on this side. There's the spindle fibers. And now look what happens. Now we get 4 new nuclear envelopes forming. And each one has an unreplicated haploid set of chromosomes. And voila, there we are. So if I wanted to carry, let's see, how can I do this? If I wanted to carry this through cytokinesis and show you the colors, let's do that. See if I can pull that off. So this one has a black one and a black one. And one of the black ones has that little crossing over event, but the other one doesn't. See that? So those two are a little bit different. And then we have this one, and this one, and then the same thing there. This one has a little bit of black, and that one doesn't. Oh, I forgot to put my black in on this one. Okay. And then what's left, that took care of the crossing over, so we have some blue. We have a blue and a blue. And then we have green. We have a green and a green. Okay. So now check it out. What are these cells? Now we have 4 haploid cells. And what these cells are, what they represent, are the gametes, right? If it's a male, these become sperm. If it's a female, one of them becomes an egg. And the thing about it is the egg, there's sort of an unequal divvying up of the cytoplasm. So 3 of them end up being called polar bodies. And the 4th one, the big one, ends up being the egg. But we won't worry too much about that. Okay, so I've taken the time to walk you through this. Okay, look at what's going on. What are important things? Crossing over is a big deal. Crossing over is the single greatest source of variability among eukaryotic organisms. How about that? Crazy, huh? And we have this crossing over being carried all the way through to the end where it creates genetic variability. Darn, I hate when that happens. It creates genetic variability among the gametes. Okay. Look how they line up. What's the fundamental differences? Well, in meiosis 1, we pull apart the homologous chromosomes and put them in different cells. Okay. In meiosis 2, we pull apart the chromatids. Meiosis 2 just being mitosis happening simultaneously in 2 cells. Okay. So now let's look at a couple of things. Let's look at, now this should all make sense. Only diploid cells undergo meiosis. The products of meiosis are called gametes. One diploid cell produces 4 haploid cells. We saw how that happened, how that worked. Now here is a drawing of it. Here's PMAT1, right? And you can see they're also using size to represent homology, and they're showing crossing over events occurring. And you can see here now there's a crossing over event. It's hard to tell with the colors. They should have picked more colors that stood out a little bit better, but that's okay. And so then there's our two haploid cells. There's P1, M1, A1, and T1. And then look, they don't even draw out meiosis 2. They just go straight to the second telophase where they have now pulled the chromatids apart and made 4 separate cells. Okay. This tells you what's going on in these different phases. Okay. I left out something very important right here. What did I leave out? Crossing over. Don't forget about crossing over. I'm going to write it in. Okay, very important. Chromosomes, homologous chromosomes line up opposite each other, and then they are pulled apart. 2 nuclear envelopes form. Meiosis 2. Great news, y'all. Meiosis 2 is exactly like mitosis. So if you know mitosis, you automatically know the second part of meiosis. Okay. Voila. That's almost the end, but there's one thing that I forgot to tell you for those of you in the watching these videos exclusively. And it has to do with the fact that there are actually 2 different kinds of cells, and this would have been something that we would have covered at a different time. And so I think I'm just going to leave that aside for now. Okay. So that ends this discussion. I hope it's been helpful. And this actually ends the unit as well. This video ends the unit. So be aware, we are just about ready to have another test. All right, team. Have a great day.